The life cycle of a bird requires extreme physiological transitions, from growing specialized tissues to maintaining a constant internal temperature in freezing weather to timing reproduction with the seasons. A single biological system governs all of these shifts, the avian thyroid gland. Looking alongside the trachea, we find the paired oval lobes of the thyroid gland. Zooming in on the tissue, the architecture consists of distinct rings. These are follicles, formed by epithelial cells called thyrocytes arranged in a cuboidal pattern. This circular arrangement is a highly specialized containment vessel. It evolved to solve a specific, severe environmental constraint, the constant scarcity of a vital trace element. The synthesis of thyroid hormones requires iodine. In natural terrestrial environments, iodine is notoriously scarce and difficult to acquire on a consistent basis. The avian thyroid overcomes this shortage through mass storage. Looking at this cross-section of the gland, the dark cellular tissue is dwarfed by massive pink pools. These are colloid-filled lumens, acting as extracellular reservoirs. Suspended within that colloid is a massive protein matrix called thyroglobulin. It functions as a biological vault, holding the raw hormone precursors until they are needed. Almost all other endocrine glands in a bird's body store their products intracellularly, keeping only a small, localized inventory within the cells themselves. The thyroid places its stockpile outside the cells. By maintaining this giant extracellular reservoir, the bird guarantees an uninterrupted supply of metabolic control hormones. Even during a prolonged fast or a strenuous multi-day migratory flight, where dietary iodine intake drops entirely to zero, the thyroid can continue to supply the body. This diagram details how thyrocytes build hormones. The NIS importer pumps iodide into the cell, and pendrin moves it into the colloid. Simultaneously, the nucleus and ER manufacture thyroglobulin and thyroid peroxidase. TPO then oxidizes and couples the iodide onto the thyroglobulin chain. This coupling yields the intermediate building blocks, monoiodotyrosine, which has one iodine atom, and diiodotyrosine, which has two. Two diiodotyrosine molecules are linked to create the primary product, T4. Droplets of this bound T4 are absorbed back into the cell through endocytosis, digested by lysosomal enzymes to free the hormone, and released into the bloodstream. The avian thyroid is highly biased in this output. It secretes almost exclusively T4, which is an inactive prohormone. Actual metabolic activation is delayed until the molecule reaches the peripheral target tissues throughout the body. To understand how the thyroid knows when to release its stored T4, we look at this schematic of the hypothalamic-pituitary-thyroid axis. The anterior pituitary directly regulates the thyroid's output. The central thyrotroph cells produce and secrete thyroid-stimulating hormone, or TSH. In the standard mammalian model, the hypothalamus triggers this entire process primarily by releasing thyrotropin-releasing hormone, or TRH. Birds have a distinct evolutionary divergence. To stimulate the thyrotrophs, their hypothalamus relies predominantly on corticotropin-releasing hormone, or CRH. This signal binds to the CRHR2 receptor. Because this specific receptor is absent on the neighboring corticotroph cells, the signal triggers a targeted, isolated thyroid response. CRH is typically associated with the stress response. By sharing this releasing hormone, the avian metabolic engine is hardwired directly into the environmental stress axis, allowing the bird to seamlessly throttle energy consumption in response to survival threats. Once the newly synthesized T4 enters the bloodstream, its highly lipophilic structure prevents it from simply dissolving. It requires specialized distributor proteins to navigate the aqueous plasma. Unlike mammals, avian blood completely lacks the high-affinity carrier protein known as thyroxine-binding globulin. Instead, birds rely on a combination of transthyretin, which has high affinity but low capacity, and albumin, which has low affinity and high capacity. Because these proteins bind the hormone less securely than a mammalian carrier would, the half-life of circulating T4 in a bird is drastically shorter. This rapid turnover rate clears the hormone from the blood quickly. It prevents centralized bottlenecks and forces avian tissues to constantly pull and manage their own local levels of active hormone in real time. Because the thyroid secretes the inactive prohormone T4, the actual physiological power to ramp up metabolism is localized in the peripheral target tissues. This flowchart details how those tissues process the molecule. The tissues rely on a specialized family of enzymes called iodothyrronine deiodinases. They act as the molecular gatekeepers of metabolism. The type 2 deiodinase enzyme strips a specific outer ring iodine atom from T4. This conversion generates the highly active metabolic trigger, T3. Alternatively, the type 3 deiodinase enzyme strips an inner ring iodine atom. This creates reverse T3, a completely inactive dead-end molecule. By expressing either activating or deactivating deiodinases, individual organs dictate their own metabolic intensity. They can upregulate or shut down entirely independent of the baseline T4 output coming from the central gland. This diagram maps the exact mechanisms the locally activated T3 molecule uses once it breaches the target cell membrane. T3 requires specialized transmembrane proteins for entry. The MCT8 channel efficiently transports the active molecule across the lipid bilayer. The primary pathway is genomic. T3 enters the nucleus, binds to nuclear thyroid hormone receptors, and physically forces a shift from gene repression to gene activation at specific thyroid response elements. A secondary, non-genomic pathway bypasses the nucleus. T3 and T4 bind directly to integrins on the membrane, triggering rapid cytoplasmic signaling cascades like the PI3K pathway. In the third pathway, T3 acts directly on the mitochondria, altering the rate of cellular respiration. This three-pronged rapid membrane signaling and direct mitochondrial burning solidifies T3 as the ultimate cellular master switch. We can see the net result of this cellular control during a massive physiological crisis, a precocial bird executing the energy-intensive process of hatching. Hatching requires several massive transitions simultaneously. The embryo must initiate pulmonary respiration, finalize yolk sac retraction, and activate autonomous endothermic heat production. The systemic trigger coordinating all of this is highly localized. Specifically, there is a sudden programmed drop in D3 expression inside the embryonic liver. Removing this D3 enzymatic brake allows a sudden, massive flood of active T3 to pour into the bloodstream. This surge is known as the peri-hatch peak. The newly circulating T3 hits the skeletal muscle mitochondria. It triggers specialized uncoupling proteins that derail normal ATP production, forcing the cells to burn massive amounts of energy for the sole purpose of generating heat. Because birds completely lack the brown adipose tissue mammals use to stay warm, this thyroid-driven muscular uncoupling functions as the definitive engine of avian endothermy. From the initial trapping of trace environmental iodide, through targeted brain circuitry, and down to the specific peripheral enzymes, the avian thyroid gland exercises precise organ-by-organ control over the bird's survival.